so we're continuing with our Chapter 1 for our Honors Chemistry class, and in this section we're going to go over the scientific method. So what you see on this slide is just an overview, which I'm sure you've seen before. These are our logical steps going through in a sort of a flowchart form of the scientific method. So we start with a problem, we gather background information so we know our problem well. We then form a hypothesis, an educated guess about what we think might be a solution. We set up our experiment. We want to do what's called a controlled experiment. We record our results from our experiment, and then we analyze those results, oftentimes with things like tables and graphs. We draw a conclusion, and then we may publish those results for other scientists so that we can further our research. All right, so we're going to go into the specific details of the scientific method now. So let's start with stating the problem. So the problem is some kind of a question that we want to answer. Oftentimes it's based on an original observation. Our background information is going to be researching our topic. Maybe we already know a little bit about our topic, but maybe we need to know more before we can really decide what we think is going to happen. Our hypothesis, as you know, is an educated guess. It's a proposed explanation for what we think is going to happen in our experiment. It needs to be written in a way that it's testable and falsifiable, meaning we need to write our hypothesis specific enough that there's a way to support it or disprove it. So if you're putting, say, fertilizer on plants and you think this fertilizer is going to make the plants grow better, we would write our hypothesis that fertilizer X, we believe fertilizer X will allow plants to grow taller than plants not receiving fertilizer X. And that way, this is a testable thing. We know exactly what our experiment kind of in general is going to be. And we have a way of disproving it, that the plants will either grow taller or they won't. Our experiment, of course, is the test of our hypothesis. Our data, again, will be tables and graphs. We'll analyze that. And then we'll draw a conclusion. Typically in our conclusion, we're going to circle back and accept or reject our hypothesis. If our hypothesis is incorrect, we may want to revise it. Or we may want to revise our experiment. Maybe we're sort of inconclusive because we didn't design a very good experiment. So in an experiment, there are some specific variables that we're always going to have. The first one is our independent variable. Sometimes it's called your manipulated variable. And that's the thing that you're going to be changing in your experiment. You are specifically manipulating and changing that thing. And notice I put the one thing. There can only be one independent variable. If you have more than one, you won't know for sure when your results come out whether it was your variable that caused them. For example, if you were wanting to test whether a particular fertilizer makes plants grow better, and you put one plant in a big pot and one plant in a little pot, and then you give one fertilizer and one not fertilizer, and one grows better, is it the fertilizer or is it the size of the pot that changed your results? And so you only want to have one independent variable. You can only change one thing at a time. Our dependent variable, or our responding variable, this is going to be our data. Sometimes I say to think of it as dependent and data, both starting with a D. So this is going to be what we collect as a response to the change that we made. Control group. A control group is something we use for comparison. Don't ever say that the control group is just a group we leave alone, because that's not exactly what it is. It is a group that's treated exactly the same as our experimental group, which is the group that's receiving our variable, but the control group does not receive the change. So if we're testing fertilizer on plants, yes, one would get fertilizer and one wouldn't. The one that doesn't get fertilizer would be our control group. But in addition to not getting fertilizer, everything else about that plant, the size of the pot it's in, the amount of water, the amount of light, the species of plant, all of that should be the same between our control group and the group we are testing, which again is called our experimental group. So this is going to be the group that actually does receive, for example, the fertilizer in a plant experiment with fertilizer. So no fertilizer gets fertilizer, and then we would compare those results to decide if fertilizer had an effect or not. Controlled variables, be careful now that you don't mix up control group and controlled variables. Controlled variables, also sometimes called constants, these are the things that you'd want to be the same for all of your groups. So in our little fertilizer experiment, we'd want to have the same species of plant, the same amount of light on each plant, the same amount of water given to each plant. Each plant should be in the same size pot. And so these would be all things we'd want to be the same so that our sole difference would be this dependent variable that we were going to be, the independent variable that we were going to be changing. And that way, when we take our measurements, we would know for sure that it was the independent variable that caused that change and not some outside factor. Now, here are 3 sample experiments. We're going to walk through these very quickly. I did this in class. And just identify those different experimental parts to make sure everybody's okay with that. And then we're going to move on. So, experiment 1. A doctor wants to examine whether a new drug increases the maze-running performance of older rats. Just like aging humans, older rats show signs of a poorer memory. Dr. Smith teaches 2 groups of older rats to find a piece of tasty rat chow in a maze. One group is given the new drug while they are learning the maze, and the second group is not given the drug. 1 week later, having learned the maze, he retests the rats and records how long it takes them to find the rat chow. So, first, our independent variable. Our independent variable is going to be what he's going to change. So the only change between the 2 groups of rats is the drug. To give the drug or not. That's really our independent variable. That's the thing that we are changing. I'm just going to put IV for independent variable. So that's what we're changing. Dependent variable. What are we measuring in this experiment? So in this case, we are going to be measuring their maze performance. Notice one group's given the drug, one isn't given the drug, and then we're measuring how long it takes the rats to find the rat chow. So that would be our dependent variable. I'm just going to put DV for dependent variable. So we're changing whether they get the drug or not. We're measuring their time to solve the maze. Our control group, that's going to be the group that is not given the drug. They're going to be exactly the same as the other group. I'm just going to put CG for controlled group. And then our experimental group will be the one that is given the drug. So this would be the drug here, this group here that's given the drug. This would be our experimental, I'll just put EG for experimental group. I don't have a lot of space here. All right, and then constants. You could probably come up with several constants on your own, but one would be they are all being trained when they're given rat chow. They should all be doing the same maze. They're probably all older rats. So all of these would be things that we would consider to be constants, things that are kept the same in all of our groups. All right, experiment 2. A researcher wanted to study the effects of sleep deprivation on physical coordination. The researchers selected 25-year-old male college students and deprived some of them of either 24, 36, or 45 hours of sleep. One group received normal sleep. He measured their ability to complete an obstacle course after the period of sleep deprivation. All right, so first we'll start with the independent variable. That's going to be what he changed. So the one thing that's different among his subjects is going to be the amount of sleep deprivation. And that's going to be our independent variable, the thing that he is varying or changing. Dependent variable, what is he going to be measuring? Well, it says that he's going to measure their ability to complete an obstacle course after they've been deprived of sleep. So this would be our dependent variable. This would be the data that he's going to collect. Our control group would be these people, the ones that got normal sleep. Control group. They are unaffected, and they're going to give us sort of a baseline of how quickly somebody could complete the obstacle course under normal conditions. And then our experimental group would be all of these people. So this would be our, I'll just put EG, our experimental group, because they're all being deprived of different amounts of sleep. And then finally, our controlled variables, things that we would want to be the same. Well, they're all 25-year-old male college students. That is specifically given to us. We might also assume that we would want to have the same obstacle course for all of the people. Maybe that this is conducted in the same location, the same time period, etc. All right, our last one. An investigator had 60 subjects watch a videotaped reenactment of a bank robbery. Half of the subjects were asked by a police investigator to recall the event, while the remaining subjects were interviewed by a police investigator while they were hypnotized. The investigator measured the accuracy of the description of each group of subjects. So our independent variable, the one thing that's going to be different, would be whether they were hypnotized or not. I'm just going to put hypnotized or not, and that's going to be our independent variable. That's going to be the thing that the investigator was changing. Dependent variable, he is measuring the accuracy of their description. So this would be our dependent variable. He's going to see if hypnotized they have a better or worse ability to recall what they saw on that videotape. Control group, that would be the subjects, I would say, that are just asked to recall the event without watching the videotape. That would be our control group, because they're not getting anything special. They're doing it the way that you would kind of assume that everybody would do it. And then the group that was hypnotized, just that group, that would be our experimental group, because they're getting something different. So just to clarify, because it's pretty sloppy and I apologize, the independent variable would be whether or not they were hypnotized. That's what's being changed. Within that, the experimental group is the group specifically that was hypnotized, and the control group is the group that was not hypnotized. And then controlled variables. I would assume that maybe the 60 subjects are similar in age, maybe similar intelligence. They're definitely going to watch the same videotape, and I would recommend that they're interviewed by the same person, because that might have an effect as well. And I'm sure you could come up with some other things. So I hope that kind of gives you an overview of the scientific method. In our next lecture, we're going to specifically continue on and talk about the data portion. We're going to talk about graphing and then analyzing and drawing those conclusions.